All right, now we're recording. So when we speak now, things will be recorded, so be careful. If you want things for posterity, please do so. All right, I'm going to go ahead and start the seminar now. Today for the Manter Laboratory of Parasitology seminar for noon on the 10th of October, it's 10/10/24, which is kind of neat. You don't get the date mixed up with the month on this one. So where should it be? Is it 10/10 or is it 10/10? Anyway, so today we get to hear from one of our senior research fellows in the Manter Laboratory of Parasitology, Dr. Karl J. Reinhard. And Karl is going to talk about 40 years or 44 years of research in paleo and archaeoparasitology since basically his beginning here and probably even before at the University of Nebraska, Lincoln. So Karl's broadcasting from home today. We also have a special guest from Ukraine, Vitaly Karchenko. Thank you for being here, Vitaly. And as other people come in, we will allow them in, but thanks for being here, everyone. So Karl,
to see you.
Very good to see you, and we hope to see you very often for a long period of time. All right, Karl, go ahead and you can take over the screen just like on the Twilight Zone.
Okay, here we're going to give a try to share, and then I'm going to go here. All right. Are you seeing my presentation?
Yes, it looks wonderful.
All right, then I'm going to make everybody disappear. View. Hang on a second. I'm trying to make make you guys disappear. I think what I want to do is.
I think we're small enough.
Yeah, you're taking up my entire screen. Okay.
We are?
Yeah, it's my.
Looks good from here.
You're muted.
Oh, Karl, you're muted now. Yeah, you're still muted.
Putting on the audio.
Okay, now you're back on.
Can you see me?
We can see you, but your presentation disappeared into the ether zone.
Okay, so here we go. Now, let's see. Zoom always makes me a tad bit nervous. All right, so I'm going to go share, and then I'm going to go do this, and then I'm going to do that. Share. Oh, how is it that we were doing fine? Yes. All right.
Excellent.
So we're going to go back in time to when I first started taking up the exploration of parasitology through coprolite analysis. And I began in 1981, testing some hypotheses about parodic hyperostosis and iron deficiency anemia that were prevalent at the time. And one of the main researchers exploring the cause of bone pathology was Mahmoud El-Najjar. And El-Najjar put together a whole series of papers on maize dependency and parodic hyperostosis, which are bone lesions in the cranium, and proposed that they were due to diet, not parasitism. So when we look at the lesions of parodic hyperostosis, they consist of a thick porous expansion of the posterior of the skull. Those are the parietals, which you see on the left side. Another pathology could be confused with it. It's called cribrorbitalia, which consists of thickened porous areas on the orbital roofs, which you can see on the right. Now, El-Najjar proposed that the variation he had found in parodic hyperostosis, which he interpreted as being due to iron deficiency anemia, related to different access to resources between Sage Plain people, ancestral Puebloans, who lived in environments such as a plain shown on the left, as opposed to canyons, which and canyon bottom sites, which is shown on the right. So he proposed that at in the 1970s, people did not widely believe that parasites were a problem for prehistoric North American populations. And he specifically said, we have to look for a dietary cause for these lesions because parasitism was rare. And you can tell that this was at an entirely different time because he was free to analyze crania, which he would not do anymore. So that what he determined was that there were these two dichotomies with regard to environment, that there were sage bottom sites or sage plain sites and canyon bottom sites. He believed that the canyon bottom sites were heavily dependent on maize, whereas people in the sage plains had a great variety of wild animals and vegetable foods. Mute
Hey,
my.
Carl, you're muted again.
Can you hear me?
Now we can.
Okay. I'm going to move this thing way over there. Okay. So he has picked out 3 canyon bottom areas: Inscription House, Canyon de Chelly, Chaco Canyon. We'll look at some of those sites later. And then 2 sage plain areas, the Navajo Reservoir and the Gran Quivira. Now, uh-oh.
I've been to all those places.
Me too, oddly enough.
I was catching mice there in those places.
Now, here he describes the diet as he interprets them for the Canyon Bottom folks and the Sage Plains folks. Canyon Bottom is predominantly maize with squash and beans. Animal food is scarce, which he uh, interprets for both Inscription House and Canyon de Chelly. And then for the Sage Plain sites, he has an abundant variety of foods with many species of fish, animals, birds, deer, and antelope, but also cultivated corn, beans, and squash. So that is what he was believing at that time. Navajo Reservoir today is a recreation lake. Navajo Reservoir in the past was a fairly barren and foreboding place. This picture comes from a site called Echoes from Eden. And I'm just giving credit to this person because all the other pictures that are not mine are National Park Service. But this shows the sage plain of Gran Quivira. And Gran Quivira itself is a complex historic mission site with a variety of small pueblos surrounding a big religious complex. Inscription House, he believes, is a classic canyon bottom site. There is a canyon of Navajo National Monument. There is a large alcove, natural alcove. And in this picture, you can see the buildings of Inscription House constructed in that alcove. Now, he predicted that parodic hyperostosis, if it's due to iron deficiency anemia, would be more common in populations with diet consisting of maize. And individuals with high iron needs, like women and children, will display more of the lesions of parodic hyperostosis. The distribution of parodic hyperostosis is certainly, let's look at children. The percent with parodic hyperostosis varies between 63.6 and 88% of the skeletalized children from canyon bottom sites, but just 15.9 to 18.2 from sage plain sites. That's pretty interesting. and he believed that it was due to diet. Statistical significance was demonstrated for the difference between parodic hyperostosis, i.e. anemia, between sage plains and canyon sites, but also he found that the variation between children and adults with regard to parodic hyperostosis is also significant. So many more children exhibit the lesions than adults. The findings, the data that I just presented from his work shows that people in the sage plains had less parodic hyperostosis, therefore less iron deficiency anemia. In contrast, the canyon bottoms were heavily dependent on maize, and the more parodic hyperostosis was seen. Therefore, he interpreted that parodic hyperostosis was due to maize dependency, keeping in mind that at his time of research, parasites were pretty much unknown for the ancestral Pueblo people on the Colorado Plateau. So my 1984 thesis and my 1989 dissertation, I tested the foundations of the maize dependency hypothesis. Was there really variation in maize between various sites in the Colorado Plateau ancestral Puebloans? Was meat more common in sage plains diet? Was there variation in meat? Was parasitism really rare? And another question I had is, was maize dependency an actuality? In other words, did people eat maize and nothing else but maize, or were there a number of other plants in their diet? I thought that would be an interesting thing to ask. Well, microscopically, there's no difference between canyon bottoms, remains of maize, i.e. kernels and seeds, between the two groups. Therefore, one could conclude that maize dependency was not more pronounced or less pronounced at these different areas. I found over the years that maize pollen is a good indicator of maize consumption's reliance at archaeological sites. With regard to maize pollen comparison, I could not find a statistically significant difference in maize between the two areas. With regard to meat, and percentage-wise, there's actually more evidence of meat in coprolites, and this is in the form of hair and skin and scale, and small animal bones and large animal bones in the excuse me, I was distracted by a note that this meeting's being transcribed. Okay, good for us. I'm going to press the OK. Okay, percentage-wise, there's more evidence of maize in the canyon bottom sites than the sage plain sites. However, statistically, this wasn't significant. But what does mean that people in the sage plains had just as much meat as in the canyon bottoms. So essentially, the maize dependence hypothesis was disproven, but was not supported by my work. Now, parasite prevalence did exhibit highly significant variation between sage plains and canyon bottoms. So 31% of the coprolites from canyon bottoms contains some evidence of a helminth parasite. Importantly, we've gone from, this is the 1980s, in 10 years, being skeptical that parasites were present in the Colorado Plateau at all, to having to accept that they were very, very common. Now, if we get just to pinworms, which are primarily what we found, pinworms were abundant in the canyon bottom coprolites. So this is Enterobius vermicularis, and quite rare of the sage plain coprolites. So therefore, we have a difference in parasitism between the sage plains and the canyon bottoms, and the canyon bottoms have the most parasites. Canyon bottoms also have the highest prevalence of lesions and crania. Therefore, it looks like parasitism is associated with iron deficiency anemia, as interpreted by El Najjar and his colleagues at that period of time. So, our conclusion, as just what I said, maize and meat were eaten in all environments equally, but it is the parasitism that was significantly higher in the canyon bottoms. This graph haunted me since my dissertation, and this is why. At the time of my writing in 1988 and the publication of my dissertation in 1992, the clinicians have found no significant pathology caused by pinworms. And so my interpretation of all of this is that what we're seeing in this this regression is that higher parasitism or higher parodic hyperostosis prevalence or deficiency anemia is predicted by the higher prevalence of pinworms and coprolites. Now, I said that's not a causal relationship. I said probably pinworms are a proxy gauge of general infection because there's so many routes of infection that pinworms have, from retroinfection to airborne infection to inhalation. So that is how I left my dissertation. Now, why was I haunted by that graph over the years? Well, for one thing, I caved into a clinical perspective. I discounted and underinterpreted my own data because clinical parasitologists didn't find pathology in pinworm infection. And my lesson for myself was to keep my faith in a great regression with statistical significance. That is as good as gold in science, at least the science that I practice. Now, research reinitiated in this topic when I met Morgana Camacho in the School of Public Health and the Oswaldo Cruz Foundation, who encouraged me to work with her in returning to this topic in the Southwest with parodic hyperostosis. So there is Morgana, our Morgana and I in the field, and then there we are in a laboratory at Aztec Ruins, sorting coprolites. Now, what Morgana impressed on me in our early meetings is that pinworm has been related to iron deficiency anemia. And ectopic infections are common, occur in many locations in the body, and some of the infections are predictable, but some of them are just quite surprising. So the revelation that pinworms cause pathology was important to me. And as Morgana and I went through the literature, we found more and more evidence, both from case studies and then epidemiological studies, that pinworm could be a serious pathogen. The difference between the 20th century and the 21st century is we're getting more parasitological studies from the developing world, where population density and decreased sanitation on a personal level results in higher pinworm outbreaks. And as we often discussed, pinworms in favelas in Brazil, that level of pinworm infection is similar to what we were finding in the ancestral Pueblo coprolites. In other words, if we look hard enough, we can find analogs for prehistory in the modern world. So this graph shows the first four case studies, or first four examples are case studies. And then we start seeing some really nice epidemiological studies with Olivares originally in 2002, but with more accumulating into 2022. So we know from these studies that iron deficiency anemia and vitamin B12 depletion is associated with pinworms. The ectopic complications are, for me, astounding. But why is it astounding to me? Because from the prehistoric perspective, there was no sophisticated surgical intervention to assess and correct these problems. So when we find these fatal peritonitis cases, as we see in the case in the jejunum and also the two cases in the bowel, when we see perforation with acute peritonitis in the small intestine, necrotic cementum in the jejunum, we're looking at pathology found today that, at least in most cases, except those fatal cases, could be addressed, but these cases would not be addressed in prehistory. Therefore, I am astounded by the potential of ectopic infections for causing issues in prehistory. And it makes me think that prevention of pinworm infection must have been a major goal of ancestral puebloans. Pulmonary migration, the parasites have been found, or pinworms have been found in the lungs. They've been found in the spinal cord and central nervous system. Amazing to me. And the less amazing are the migrations to the urogenital system. And this has been a major focus of recent research with gynecologically focused clinicians. And I think this opens up the potential. I hope Ida is listening, for working on a paper on prehistoric female health and pinworm infection. And of course, the unusual cases are always the eye and the nose. Okay, the lessons that I have accumulated from this is, or are, if I hadn't discounted the validity of my data due to the clinical perspectives in 1992 when I was writing my dissertation and publishing it, my study would have been a portent of the future of clinical findings. In other words, my study from ancient times would have foreshadowed research from the clinical sector in the 21st century. I lost out because I was shy. As far as the regression, I've had worse regressions and published groundbreaking papers. So now I'm cursed by an unavoidable regret at not having captured the day at the time I was doing this work and really moving science and medicine forward. Regret. However, working with Morgana and from 2013 to 2020 and writing up until today, we defined it for the Colorado Plateau extreme pinworm parasitism on a chronic level. So what have we found? One thing we need to remember as we go through the rest of these data is 5 to 10% of modern pinworm parasitized individuals pass pinworm eggs in their feces. That means that 95 to 90% of infected people are not diagnosable through fecal analysis. Wanted to point out that these are the percentages of pinworm infection from various sites that I found during my dissertation workout or just research. And as you see, some sites with prevalences from 10% to 25% really represent ubiquitous infection risk, because those prevalences pretty much represent 100% infection for these populations. Now, Morgana began, and also Jonica Morrow began a new method of study. The analysis that I just showed you before were my analysis in which I was looking for egg concentrations greater than 600 eggs per gram. I was looking for symptomatic cases. Some other researchers whose data I include were simply looking at the data from a presence absence perspective. However, with these researchers, we began looking for all infections. And when we began looking for all infections, we feel confident that we found all the infections for these sites. So these were Morgana's dissertation sites. Today, I'm going to focus on Aztec ruins and Solomon ruins. Excuse me. I'm going to get a drink. All right. So one of the things we did is refined our methods and recovery of parasites from mummies and coprolites. An epidemiological approach was published in which we described new methods for capturing all infections in a coprolite analysis. And so when we applied our new methods, which are in bold, you could see that with excellently preserved coprolites, we found many more cases. So as opposed to these old data in which I was looking for symptomatic infections, we can see many more infections that I missed simply because I wasn't concerned with background infections. Even when we look at poor preserved coprolites, with the bold studies, we're finding more infections, which I think indicates a success in applying new approaches. Please note the difference in positivity between Aztec ruins, room 225, New Mexico, and Aztec ruins, room 219, New Mexico, between 73 and 14%. Diet and parasitism. Okay.
Isabel Teixeira Santos, Morgana Comatosis. We decided to.
I have apparently, can you hear me?
Yeah, we can hear you, but something happened. So you try to hit play again and see if it works.
I think that this has been recorded, and I've forgotten how to slideshow. I think I take that off. Oh my God.
Your God or mine?
Sorry.
Yeah, suddenly we had two Carls speaking, which is pretty cool, but
Well, it would be great. It bothers me quite a bit though. So what I need to do is go to my presentation. I'm going to mute myself if I can.
okay. So pretty interesting stuff so far. Yeah, so one of the things that Carl and I have been working on since I was a graduate student is trying to get DNA from these things. Just to let you know, you're still muted in case you need to know.
we go. Can you hear me?
Yes, we
Hello?
can hear you fine. We also see
Okay.
you.
Sorry about that. I think I had a recorded slideshow.
Doses. We
Oh
decided
my
to
God.
So it
This
says
is,
you started playing your slides.
yeah, what I'm gonna do now, you can see me advance, correct?
We can't see, all we can see is your beautiful face and your well coiffed hair.
Okay, so, um, slideshow.
Everybody is cool. Don't worry.
All right. Okay, well, what I'm gonna do is, I wonder if I can turn off the sound of my computer and, so, how do I silence my computer? I know, I do this. Okay, Scott Gardner. Can you hear me now?
Yes,
Not hearing anything? Turn up volume.
we can hear you. No problem.
Can you hear
Okay,
us?
can you hear me? All right.
Yes.
What I'm going to try to do is, I don't know how to turn off the recorded, um, clear narration on all slides. Okay, I think we could do this.
Okay.
Okay, make it big. Okay, I can make it big and I'm going to share. Going to go to Zoom. Zoom.
Ethan, you need some more books on those shelves.
Yeah, I know it's a pretty embarrassing lack of
Okay.
material.
So sorry for that. I think we're going to be okay. Can you hear me? Uh-oh. And then I go here. Can you hear me?
Yes, sounds great. Your
Oh,
voice is
great.
mellifluous.
Great, 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 great. Then let's get going. I'm so sorry. So what we're going to do is go to the Colorado Plateau, which is right here, where it says United States of America, because that's a true America on the Colorado Plateau. Now that we're there, we're going to go to this little area near Farmington, New Mexico, and there's,
We know where that is, but we can't see your screen yet.
oh, okay.
But I've been to all those places, so I have a visceral feeling of those places.
Okay, now I'm now having a difficult time. Share. Can you see it now?
See, yes.
Okay, good. Again, I'm going to apologize for being not quite as good as I am. All right, as good as I could be. Okay.
Working
So
perfectly.
what Morgana and I began doing is testing this 2009 hypothesis in the diet-based in maize, intense parasitism together affected maternal health, resulting in deficient vitamin B12 and other nutrients. Weaning foods were nutritionally deficient. That affected infant nutrition and also parasitism. Note that the parasitism comes down to mothers and infants, and that resulted in vitamin B12 deficiency and parodic hyperestosis. So, there we are, and our study sites are there along the Animas River and the San Juan River near the Four Corners borders. Now, the sites are kind of interesting because they're both used at a time of warfare. They're both have been built very early, but the occupations we have between 1140 to 1280 is a time of population aggregation. And people move to these sites because they're accessible to water. So that is a 3D reconstruction of Salmon ruins. There is Salmon ruins as we excavated. My excavation area was over down here many years ago when I was young. There is a latrine. The red dot marks a latrine at Salmon ruins. And this is a stratigraphic image of the deposits of the latrines. And note that they're layers of feces interspersed with layers of aeolian windborne sand. Aztec ruins is a large village. There are actually several large villages like this in the site complex. But in Aztec ruins, there's two latrines, room 225 and room 219. When we look at and compare the prevalence between Aztec ruins and Salmon ruins, and between Aztec ruins, we see this really dramatic difference of prevalence between room 219 and 225. These rooms were used at the same time as was Salmon ruins. So that leads us to wonder what's going on. And before we get to what's going on, the lesson I learned at this point is making up for lost opportunity, i.e. my thesis and dissertation, involves listening to colleagues, so Morgana had to convince me to return to the parodic hyperdosis debate. And then returning to a discussion that I've left required a lot of effort on the part of myself and Morgana to collect a convincing data set. So what did we do? Well, I mentioned before that if pinworm is such a big problem for ancestral puebloans, there must have been a need to purge themselves. In the process of going through rehydrating and recovering seeds from Aztec ruins to 219, we found a lot of weird things. The most remarkable weird things were leaves and also squash seeds. Now, when we look at the general perspective of food distribution between these three latrines, Aztec 219, Aztec 225, and Salmon ruins, room 62, we see that the squash seeds are way more common in room 219 than any of the others. And similarly, bean leaves are more common as well. So why is that? Well, okay, here we're doing fine. All right, the bean seed evidence is microscopic, and the evidence are these trichomes, which are what we call bean hairs in non-scientific jargon. They're resilient structures made of lignin, a plant durable compound. I compare them with a variety of plant hairs and lignin structures from other domestic plants, and only beans for ancestral pueblo plant foods have these trichomes. And they're interspersed with other dietary foods like maize pollen is represented in these structures, and other leaf tissue, these tracheids, these little fibers that go through leaves. Imagine you're eating celery, and that fibrous stuff is microscopically is composed of these little spring-like fibers. And these trichomes are what signaled to me that we have something interesting going on. So squash seeds and bean leaves, and the bean leaves characteristic are covered with trichomes. Well, as we all know, researchers working with chimpanzees in various parts of endemic areas have discovered that leaf swallowing is adaptive to controlling a variety of nematodes. 4 different plant species are used, and as you can see, all of them have trichomes. These are nice hooked trichomes like beans. Some are long and less well distributed, and others are short and tightly compact on the bean surface, on the leaf surface. So what happens in these cases is uh, the leaves and those trichomes together effectively purge the chimpanzee system. Hoffman and his colleagues writing about this said much is known about the function of the gastric compartments of Homo sapiens. And they say that this process of eating leaves for humans would probably act the same way as chimpanzees. So let's see what happens. So this is a drawing from this article by Hoffman. Specifically, they're focused in this paper on 2 other parasite species. Let's see what happens. So when you first eat a bean leaf, and by the time I'm done with this presentation, you will not want to eat bean leaves. Let's see if I can clear. But what we see is the empty stomach, juveniles ingested, and then also people eat the leaves. That causes a physical irritation and gastric motility and secretion, and it also causes the opening of the pyloric sphincter to be stimulated. And then further down the intestinal tract, there's increased motility and secretions in the small intestine that lubricate and push the parasites downward. And as we get towards the lower intestine, the removal of the adult worms is aided by the physical surface of the leaves, which also causes irritation and localized postural contractions and peristalsis and antiperistalsis. It reminds me of a closed washer. The net effect is that the larva, the juveniles, the emergent juveniles, and adults are pushed out of the intestine. Transit time is decreased from 24 hours to 6 hours from mouth to anus. And this is important, that adults trapped in leaves and diarrheal deposits are not seen in diarrheal deposits without leaves. So it's conclusive evidence that the leaves themselves cause the excretion of the parasites. Do we need chimpanzees to tell us that? It's helpful. But at a different site, Antelope House, in the 1980s, I found a fragment of a pinworm, a female, that was deposited in there you can see the eggs in her, was deposited in a sample that had bean trichomes, bean leaves in them. So I think we have really good evidence that bean leaves were helping to purge pinworms from the gut. Now, as far as anthelminthics of squash seeds, there are a variety of articles that indicate that squash seeds, both pumpkin and squash seeds, have nemacidal potential and affect the survival of juveniles. Pumpkin seed is an anthelminthic. Squash seeds are the same species used by ancestral Puebloans. And it's a low-cost and harmless way of getting rid of parasites. That's good to know. So from the lesson of this stage of my research, discovering a community of people who are purging themselves of the parasites, I would recommend that to rejoice in the unexpected and follow the path enigmas lead to. So for us, when Morgana and I were processing the coprolites one day, the enigma were all of the seed fragments from squash and leaf fragments from beans. And eventually that that led to what I think is conclusive evidence of people struggling with their pinworm infections and purging them. This comes on like the 40th anniversary of my first anthelophantic paper, which I published in 1985. And I think this strange discovery has led us to some pretty definitive evidence. And I would also say, keep in touch until a topic resolution is reached. I could have given up interest in this altogether. Morgana compelled me to continue working on this topic. So some decades later, after I started looking for anthelminthics and coprolites, somebody had excavated samples that were able to better support what I'd begun in 1985, and that is demonstrating that prehistoric people had anthelminthics that we could find and describe later. I'm convinced that important lessons are frozen in the past, and we will find them if we're just persistent in our goals. All right, we finally are getting to the newest paper, I suppose, and this came about because of the parasitology seminar 4 years ago, in which Scott said, wouldn't it be nice if somebody looked at a bunch of coprolites through the series of at a time of climate change, so that we had before the climate change, during the climate change, and after. And I thought really carefully to myself and realized that that was the goal of my dissertation, but I never had time to explore that. So we're going to Antelope House. So here we're back in the Four Corners area. There's the United States of America, and Antelope House is here. That square is not that big. Antelope House is located in Canyon de Chelly. So it's obviously a canyon bottom site. And this is what the ruin looks like today. This little row of rooms right at the back, this is the first series of rooms that were built. And over time, this village expanded greatly. And it expanded at a time, I'm going to see if I can move this. Okay, press this. Between 1150 and 1270, that we had a period of environmental perturbation. I don't know that. Okay, so 1150 and 1270, we see that there was there were droughts at these periods of time that affected the number of habitation sites on the Collar and Four Corners area. So we're looking at a mega drought between these areas. Spatially, Native Americans were distributed across these areas marked in blue pastel and yellow pastel. At the peak of the 1150 and 1275 contraction, these population moved to where water was most manageable, most accessible. Antelope House, by virtue of the fact that it is at the confluence of two rivers that come out of the Chusca Mountains, there was water during this contraction, so people could move into the area. So what we see is dwellings above ground, adjoining rooms of stone. Larger villages were established. And then when we get towards a drought, we find that we're dealing with large settlements around water sources, isolated water sources. Antelope House itself had 2 rooms in it in 825 to 900 AD. 7 rooms were constructed in 900 to 950. Then nobody lived at the site until 1100. 22 rooms are built between 1100 and 1140, and kivas are ceremonial structures and 2 ceremonial structures. And then between 1140 and 1200, there was a multi-story construction and a plaza built with 2 distinct room blocks. 42 rooms were used with 3 kivas. And then in the latter stage, the late Pueblo III, the village was modified and reorganized and renovated to handle a large population. So I'm going to skip through these, but essentially, this is a first room block that was built at 1100. By 1270, when the site was abandoned, all of the other room blocks had been built. And I'm going to, since I lost so much time fixing my presentation, how embarrassing. I'm just going to point out that there was the north room block was built and then was remodeled to have a lot more living space and ceremonial space. So parasitism, let's focus on parasitism. Here are some parasite eggs as we find them in coprolites. And so, as I mentioned, antelope house analysis is focused on identifying pathogenic infections of 600 eggs per more or more. The biology of pinworm is such that their eggs can be transmitted by air to food. We can inhale the eggs. We can also contaminate ourselves from scratching our anuses at night when the pinworms are causing irritation, and then transfer those eggs to our loved ones later when we wake up in the day. And then there's retroinfection, in which nocturnal egg laying results in retroinfection, where the eggs hatch and the larvae re-enter the host through the anus. Now, here's some data. The minimum sample size we can use for comparison is 18. So we see from our data that for the early Pueblo 2, early Pueblo 3, which is essentially pre-drought, we have 56 samples. 18% of people had symptomatic infections. Middle Pueblo 3, we only have 13 samples, so that's a frustration. 15%, but we can't use it for comparison. And then when we get to the late Pueblo 3, we have 97 samples, good sample size, and 45% of those samples represent 600 egg per gram or more infections. So the data here indicate that at the maximum time of drought, there was a 45% potential of having a symptomatic infection at this site. So the antelope house parasitism conclusion, antelope house shows a increase in infection during the final crowded phase. The prevalence is the highest count for symptomatic infections, and the increase in infection correlates with dietary shifts, showing adaptations to changing environments. Dietary shifts are simply represented here, and that from before the drought to during the drought, the amount of fresh maize declines. The amount of dried maize, stored maize, increases. Beans decrease significantly, and gathered foods, starvation foods, are double, more than double. So that starvation foods include these kind of wild seeds, more wild seeds, and cactus. So cactus pads, which I don't show here. So what we can see is that only 9% of the pre-drought foods could be classified as wild foods. Oh, that's sad. My percentage collapsed. Okay, so 9% of the foods were gathered during the early part of the drought or the construction phase. 8% had starvation foods, but during the drought period itself, 20% of the coprolites were composed of famine foods, and many of those coprolites had no maize at all. So, conclusions, concentration of a population is associated with a remarkable increase in crowd disease due to drought and people coalescing around permanent water sources. Evident decline in the quality of nutrition. Together, these factors association with mega drought implies poor health. So I think we can say both parasitism, inadequate nutrition combine to influence the pathology we call parodic hyperestosis. Now, what are the lessons from all of this, especially this last segment? I collected those data in the 1980s and forgot about them until Scott reminded me about it in 2020. So the lesson there is a good database, if you've been really thorough and methodical, maintained your methods, it has a life that outlasts the collection date. So that good data never go bad. In an overall picture of this whole presentation, we can learn that lessons from the past in archaeological samples can relate to big picture problems of today, like climate change, parasitism, and nutritional balance. All right, that's the end of my presentation. And even with the error in having a recorded presentation, I'm still at 53 minutes. Thank you very, very much. I'm going to escape. I'm going to stop share. Here I am.
Thanks, Karl. That was truly amazing and significant.
Yeah, thank you guys.
And I'm just wondering, even from down there in hurricane-stricken Florida, Sebastian is there. Wow. So I have a question. Usually here in places with pretty good healthcare checking, we usually see pinworms mostly in kids. And do you think that we're seeing everyone infected with pinworms in these pueblos? Or do you think that a lot of them a lot of those records are from a lot of pooing from kids? What do you think about that?
I wrote a paper in 2017 addressing methods of coprolite analysis and how important it is to make sure to essentially brush the contamination off your sample. Some people will core the sample. Some people slice them lengthwise and then use just the inside of the coprolite. So those are two approaches we have to eliminating contamination. I do believe that the small percentage can be contaminated when beetles, and specifically spider beetles, get into coprolite deposits when they're dry and bore holes into the sample. If dust from the site is contaminated with pinworm eggs, then there's a possibility that small numbers of pinworm eggs enter the sample. So if we're careful, and I think most people are, we can reduce the potential of contamination. However, I think that we can wind up with about 2% of the samples being contaminated. And that's it's not only drawing a number out of my head. We found some contamination, some pinworms and animal coprolites from Zape.
Oh, right.
I would say there's a few, like I would, a small percentage that are contaminated. Then one has to ask the question, how relevant is that contamination to any conclusions? If we're doing excellent methods where our application of excellent sorting techniques result in a robust data set that will essentially overshadow what contamination we might have. Did that make sense?
Yeah, but I
Okay.
wasn't really asking about contamination. I was wondering about kids versus adults.
Oh, well, now you're getting into a very interesting question. At this point, it's really, I'm just going to say, we can tell who is laying the coprolites. However, it's more expensive than we want. So far, the analysis of excreted hormones indicates that women, females, are using the sites. If we can take the $10,000 per sample and apply it to gut microbiome work, we can identify children.
Ah,
So we can spot, we know that a lot of these are deposited by women, and some are deposited by children. And as far as whether we can identify child coprolite or an adult coprolite, I do not know.
okay, interesting. Basically, because I was thinking most of the coprolite or most of the people who are infected with pinworms in North America are most usually kids, but maybe it's because we just don't check adults. So it could be that we don't check
Hmm.
and we don't have symptomatic symptoms, and therefore we don't know. So I'm not sure what that is. Anyway, so
If the people are infected, just the way the female is laying the eggs, it must be symptomatic. There is no way that an adult is not going to say, "Hey, my anus itches," because it is the mucus of the female that serves as a matrix for the eggs that actually causes the problem.
And
Like
I
even
think
if it is one.
that's an important point, because I think that that is a symptom that lets the population, the community, know that pinworm is present in the community. And I think it may be those basic symptoms that influence their decision to go to that really drastic purging of their intestinal systems. The squash seeds and the bean leaves, that is not a comfortable way to get rid of parasites. And the other thing that I want to go back to is we actually look at the diameter of the sample to assess whether we have an adult or not.
right. Cool.
And so, any other questions?
Well, there were two things that you mentioned, Carl, but then one is about co-infections. Like basically, if they had co-infections with trichuris, with whipworm, then basically like the level of detection with the whipworm is more efficient should those whipworms be there. And at least in Sape, there were a few that were infected with whipworm. But then in that case, kind of like the conditions in which people live, then they make them kind of like prone to co-infections, right? Because they are defecating in the same cave. Then if the eggs of the whipworm are around, then co-infection for a helminth is going to be like that.
The perplexing thing for me is 10 samples that we'd analyzed. That was my first graduate student's analysis of Zappe in the 80s. In addition to all the pinworms, they are also finding whipworm, ascaris, and ancylostoma. You found whipworm and you found ancylostoma, but I don't think we've ever found ascaris. However, this is a fascinating thing. Eliza, oh, I'm getting excited about Zappe. I should never bring that up. Eliza did part of her dissertation analysis of the sediments in the earliest levels of that cave, and they have ascaris, trichuris, toxicara as a dominant species at that level with pinworm. Yeah, so I think that we may have more parasites that are cryptic, because people were very efficient at getting rid of the geohelminths. I think pinworm may be because of retroinfection and inhalation and all of that complex, very intimate system of infection may have had a more difficult time getting rid of pinworm.
Well, also we mount an immune response to all of those other nematodes because they have a tissue migration, or in the case of the whipworm, they embed themselves in the mucosa. So because of that, basically, one might expect that the prevalence could be lower, or at least the detection, because then people might, if it is adults, people might have just like, mount an immune reaction. There was another thing that you mentioned, Karl, that it is very important. And then actually in the last ASP meeting in Denver, I went to basically them to basically tell them about the data. What is it going to happen with all of those data sets? Because I told them and then I challenged them. Yeah, you are generating all of this data. I mean, we are generating all of this data. This data is basically accumulating in a notebook or in my laptop. But then what is it going to happen if all of a sudden this building catches on fire? What is it going to happen with that data? And then who is going to use it? And then what is going to be the value of all of that expertise and effort that was set forth to basically put it forth, making it possible? Like during all of this description, how many experts you're talking about? Like basically Sadauto, Morgana, Elisa, Jonica, Dr. Reinhard, and it's like a lot of like time and people who really know these organisms that shape this understanding. And it's the call that you were making to yourself, kind of like, oh, I didn't catch this thing on time, but it's like, hey, those things happen. But then what is it going to happen if that data is going to be, say, if all of a sudden that data passes on to me, and I am like, yeah, it's here, it's secure, but then my question to you is, what do you think it should be our responsibility with those data sets?
Well, one of the things that bothers me is, what if I had retired before Scott reminded me that I did that analysis that never published?
Right.
It would be gone. It would absolutely be gone.
Yeah, so it would be gone. Plus, he's going to put all of his remnant stuff into the lab here before he finishes off.
Yeah, so that my lab books and everything will go to the Manter lab. And that's one way of keeping it. But your question, and we're still being recorded?
Yes.
Your question, I'm going to see if I can say this carefully. Your question is particularly relevant to the data loss that has happened in labs in which senior members died, in which the dreams of having a data image data system documenting every egg, every larva, every found, that dream never came to pass. And so I think the best thing we can do is to take our data, pass them on to the Manter lab, pass the samples onto the Manter lab, and at least I should probably persist a few hours a week to help with any questions or archiving information that needs to be archived. My concern is I'm 70 and I'm finding every year it's harder to focus. Every year it's harder to grab words to write or communicate. And I think that I should really get this collection and the data organized so it can be used, because in 5 years, I'm just going to be worse, I think. And in 7 years, I'll probably be relaxing on a beach, learning how to drink a mint julep or something like that. But the bottom line is, older folks become less energetic, less focused, and then they can die suddenly. So these things have to be resolved. Can you and our colleagues in North Carolina do a presentation on the biodiversity of Zape for this seminar?
Well, maybe, but then when we kind of like put our kind of like more coordination, we need to chat more. But now I'm going to press on this thing a little bit more, if you allow me, because I'm
Okay.
51, and to me it is hard to concentrate every 15 minutes. Every kind of like people come with like crap that I don't really want to hear, but then I need to solve according to them. So this level, and then also I might die. Well, any moment I might cross. So the issue that I pressed and challenged the American Society of Parasitologists was not that we deposit the materials in the Manter lab, because that could be nice, but it is going to be of the same uselessness as it is today. What I am asking people is to go one step forward. And then most people don't actually forward these data sets because, among other reasons, they want credit. They want to receive some sort of credit.
Even
Then
after they're dead.
if that is the case, then deposit them on Dryad, and then my university has a Dryad subscription. University of Nebraska has a Dryad subscription. Submit that thing online, and then just basically generate that DOI, and then these people using that data, they have to cite you. That will make them the data visible and useful. And I'm telling you this not only for this is a very important situation. I was coming to think about it because of all of those parasite community of like Oxymicteros, parasite community of Thamnophis that I've been part of, for which the data is gone. And this is terrible sad, personally for me, because number one, I killed all of those animals. Number two, I didn't secure these samples, or I don't know where they are, if they are in my office. And then number two, I don't make them visible to people. Kind of like you know, like and then the more dramatic part for me, because I'm dramatic, is that the localities where I gather that information from, those habitats are gone. They are like basically a dry, Chapala in Mexico, like semi-dry, so is Cuitzeo, and then other places, and then parts of Bolivia that are totally just like basically gone with fire and with roads that they made. End of my participation. I encourage everybody to think about that data, because then it's like today you think about it, it serves for us to kind of like come up with a table characterization of the communities of the parasites. But the important information is what parasite was present in that point. And if that parasite was there 20 years ago, that we don't know because people didn't document this, or will there be 20 years from now, which they might know if we document these things.
Yeah, so the main problem with all of this is people do not save their specimens, and they don't save their collection locality data, and then they don't deposit their specimens into a museum somewhere. So then when they retire or quit or move somewhere else, everything is lost. And so many times the parasites are not even identified properly. So therefore, unless someone can come back and look at the specimens in a collection, in a museum, then they don't even know what it was. So, you know, we've been pounding on this for how long, Augustine? You know, since 1989, 1990. Anyway.
Well,
I think we should stop now because we're over time. But Augustine, we recorded your comments. We have them here.
Oh no, I didn't remember.
Karl, this was a wonderful talk. Thank you very much. And I think it's something that we can all think about for the future. And thanks everybody for joining us. Vitaly, stay alive over there. Keep avoiding the missiles and all that.
Hi, Nancy.
Hi, Karl. I
How you
arrive later.
doing?
We'll
Nice to
everybody
see
later.
you.
And
Good to see
Sebastian,
you
thanks for
too.
sending your image of the shipping routes. I'll get that into the paper. Hasta luego. Muchas gracias.
Hasta luego.
Adios, ciao.
Bye.
See you later.
Bye-bye.
Bye.
End meeting for all.